So what we're going to be talking about today is the human microbiota. A lot of times we say the word microbiome, but most of the time we mean microbiota. So again, microbiota is all of the organisms living in and on our body. When we talk about microbiome per se, that's actually the genetic makeup of those organisms. So we can specifically use that term in other cases. So looking at the microbiota, we're going to be looking at the symbiotic relationships between organisms within our body, how they help us. We'll also be looking at dyssymbiosis when we see a state of unbalanced nature between microorganisms. Okay, so some of the topics we're going to be looking at, again, is the benefits, intercommunication between microorganisms, and how we can prevent infection. So how our own microbiota will actually help prevent infection. Looking at the body, we have many different microhabitats in the body. So even just the skin itself has microhabitats. We have areas of more sweat, more salt. We have areas that are more dry than others, areas that are more exposed to oxygen, areas within the gastrointestinal tract, maybe that have no oxygen. So we're going to see a diverse group of microorganisms growing on the body and within the body. This image here highlights some of the main organisms that we may be familiar with. We're going to name a couple of them too. So we've probably heard of things like Staphylococcus, Candida, Helicobacter, Lactobacillus. Those are a lot of the common microorganisms. So we'll mention a couple of them coming up here. One thing I wanted to point out about the different habitats that we have on our body, essentially skin, mouth, genital urinary tract, and the colon, we can see a different relationship to organisms who want to live in an environment with oxygen versus organisms that want to live in an environment without oxygen. So we call this aerobe to anaerobe. Aerobe is an organism that lives in an aerobic environment with oxygen. Anaerobic is without oxygen. So when we look at our skin and our mouth, this is exposed to the outside environment. We're going to see oxygen is there. That's why we see this 1 to 10 ratio, aerobe to anaerobe. So even though we have more anaerobic bacteria, we see a higher concentration of aerobic bacteria versus something like the genital urinary tract or the intestines. So going through the GI tract, where we have a very, very low percent of aerobic bacteria. So mostly all anaerobic on the inside of the body. Okay. So let's look at the skin. Again, the skin has different microenvironments within itself. A lot of them can be salty due to sweat glands. They can be acidic, and they can be moist or dry, depending on what kind of environment we're looking at. We could see 10 to the 12th microorganisms populating our skin. So we have our skin is over 2 meters. It's a very, very large organ in our body, and it's very diverse. And the habitat, again, is diverse and supports different microorganisms because of that diversity. Moving on to the oral and nasal cavity. Again, these areas are exposed to the outside environment. So we're going to see more anaerobic, oxygen-loving microorganisms in these environments, just as we saw with the epidermis, the skin. Particularly, one thing I want to mention about the eye here is that we have a specific chemical we can produce in our eye to prevent microbial growth. So our eyes, our nose, and mouth are exposed to the outside environment. And we produce lysozyme in our eyes, which can break down microorganisms that get into our eyes. So while we are exposed to the outside world, to microorganisms through our eyes, a good percent of the time, we have this defense of lysozyme to break down microorganisms. It's also something found in eggs to prevent bacterial growth. Okay. Oral and nasal cavity, again, these are open air systems. We'll be looking more at oral hygiene, oral bacteria later. Okay, a lot of students ask, where do we get our microbiota from? So our microbiota comes from birth. So with the normal vaginal birth, this is where we first come into contact with microorganisms. So the vaginal microorganisms are the ones that colonize our skin. Whatever we come into contact with is going to colonize our skin and our body, these microorganisms. And that's where we get our microbiota. And microbiota versus your microbiota are going to be different based on what we're exposed to. Now, this can change throughout our lifetime. It can change depending on what we eat, what we're around. So there's many different factors that play into it. If we think of something maybe like a cesarean, maybe you're thinking, oh, I wasn't born, I wasn't a vaginal birth, I was a cesarean birth. Well, whatever you were first exposed to would have been the microorganisms that colonized your body. Okay. Oh, got a graphic picture coming up. Um, looking at Looking at microorganisms, we know microorganisms can move from one place of the body to the next. Looking at dental procedures, if we get cuts in our gums, if we're breaking off plaques of bacteria, we know that we said we had all of those, we have all those biofilms building up on our teeth, those plaques. If they break off during a dental procedure and that gets into the vascular system, into the blood system, it can move to the heart and it can take on a vegetation where it grows on the heart, and that's what we're seeing here. So it can be very dangerous. Okay. Looking at the respiratory tract, we can see different types of microorganisms, again, living in the respiratory tract. This is an open system, so oxygen can go in and out. We have a ciliated lining like this that we see here. This is a scanning electron microscope image that is pseudo-colored. With this ciliated lining, we can actually look at the mucociliary escalator, which is one way that we prevent bacterial colonization. So as we breathe in with this open system, we're breathing in microorganisms, and our mucociliary escalator, like when we cough, when we push air up, these little filaments, these little ciliated filaments push microorganisms back out of our body. Something that's interesting for people who smoke cigarettes is that this mucociliary escalator is actually slightly disabled with nicotine. So as people smoke, they're unable to get rid of specific microbes that they would normally cough out because they have paralysis of the mucociliary escalator. So sometimes people will wake up and be coughing, then they'll start smoking and they'll stop coughing and they'll think that the cigarette possibly made them better, but it's because it actually caused a temporary paralysis of the mucociliary escalator and can still be causing harm. Okay, moving on, let's talk about the stomach. The stomach, as we know, is an acidic environment. Acid is mostly lethal to bacteria. So as we bring in food from the outside world into our body, if it comes into contact with the stomach through the gastrointestinal tract, the stomach can kill off quite a bit of bacteria. So we talked about leaving our food out. Every 20 minutes we have microorganisms growing. Well, for a normal amount of bacteria, we can kill that bacteria within our stomach. So it's not a problem to have bacteria. But we don't want excessive amounts of bacteria. We don't want biofilms of bacteria because in biofilms, even the acid of our stomach can't really get into it. That's why it's important to look into food preservation, food safety. So, as we know, the acid kills off most of the bacteria within the food we eat. There are some microorganisms that actually find this habitat, this acidic environment, to be extremely habitable. So you may or may not have heard of Helicobacter pylori. That is a microorganism that actually live and thrive within the gastrointestinal pits within the stomach. So this is Helicobacter pylori that we see here, and it's integrated into these pits within the stomach. So it happily lives there, happily multiplies there. And actually, a good percent of the population is infected with Helicobacter pylori. Many people are asymptomatic, but some people do come back with gastrointestinal ulcers or gastrointestinal cancer because of this infection of Helicobacter pylori. Okay. Helicobacter pylori, another thing also, it's not as prevalent in America. We see it in other countries, specifically countries with low socioeconomic status. And maybe it's related to lower hygiene as well. So maybe not access to maybe soap or proper hygiene techniques. Okay. Let's move forward onto the gastrointestinal tract here. So a good percent of the gastrointestinal tract, again, is not exposed to oxygen. So as we move food through the stomach, we'll be focusing on the intestines here. Within the intestines, we're going to have mostly anaerobic microorganisms. Again, anaerobic means no oxygen as we're moving through the body. For a good portion of the intestines and the gastrointestinal tract, it's mostly acidic. So again, we're killing bacteria, but slightly in the duodenum, we see a slightly alkaline environment. So that's one of the only differences that we see. So that microenvironment is going to promote the growth of specific bacteria that like living in a more basic alkaline environment. Okay. Looking at the intestines, we can look at the benefits of having microorganisms in our intestines. So here we see this scanning electron microscopy image where we see different types of species of microorganisms. We see bacillus, we see cocci, all living together. So all of these microorganisms have different benefits to our body. We have mostly a symbiotic relationship with them. Some of them is that they're fermenting some of the foods that we can't ferment ourselves. So as we consume food, not all of it will be fermented by our body. It will move into our intestines, and within the GI tract, these microorganisms will pick up that food, break it down, and make more energy for us. Now, in the situation that we see in the United States, we don't normally see people lacking calories. So having additional microorganisms that yield additional calories, more than the calories that we're consuming, is not necessarily beneficial. But if we think about thousands of years ago, or if we lived in a place where we had limited food, limited resources, this additional fermentation of these microorganisms would be extremely beneficial for survival. But it's not necessarily beneficial for survival for what we see in America, where we have too many calories coming in most of the time. And we'll look at that in a minute. Also, the immune system, we may not know this, but the gut microbiota can actually help train our immune system so it can identify pathogens. Again, when we consume pathogens, when we break them down with the acid in our stomach, with the acid within our intestines, we can break them into small pieces and we can train our immune system off of those small pieces. So if we do have a larger infection with those specific microorganisms, that our immune system can identify them quickly. Okay. We have preventing growth of pathogenic bacteria. This is important because our intestines are already colonized with microorganisms. So when other pathogenic microorganisms make their way down through the digestive tract, they can't really plant. They can't really take root because there's no area for them to survive. All of the territory is essentially taken by your existing microbiota. So they would be fighting for an area to live, they would be fighting for food, and you have an existing colonization of microorganisms. Following that, we have vitamins. Some of the vitamins that are necessary for our health, we don't make them ourselves. It's actually the metabolic processes of the microorganisms that produce vitamins for us. So again, they can consume some of the food that we don't digest going through our GI tract, and they can produce vitamins for us. Another thing is producing hormones. They can actually change the hormones that we have. We'll talk later about the gut-brain barrier too. Okay, so focusing on the intestine, I want to talk a little bit about weight and our association of weight with our microorganisms in our GI tract. So here we're seeing gut microbiota and obesity. Here we see normal patients, and in red, we see a high percent of just bacteria. In obese patients, we don't only see bacteria, we also see archaea and methanogen bacteria, a bacteria that work with methane. Looking at patients who have had a gastric bypass, for the majority, most of them after the gastric bypass only have bacteria. We see some of them have the archaea and methanogens here, but in a lower percent than the obese patients that we saw here. So this makes us think that methanogens, archaea may be playing a role in obesity. So let's go look into that here. So here we see a relationship between microorganisms and between how we break down food, how we ferment food. So let's go ahead and look at this process here. We have step one, we have this hydrogen gas buildup. We have hydrogen gas buildup from fermentation in our body. And hydrogen gas is actually working back on a feedback inhibition loop. So as we ferment and we build up hydrogen gas, when we build up hydrogen gas to a certain amount, it will inhibit additional fermentation. So we will stop fermentation. We will stop making essentially more calories for our body. So our body learns to train itself. We make too much hydrogen gas, the body will stop fermentation. We don't need any more of that fermentation. We have enough calories. But again, we saw methanogens in people who may be struggling with obesity here. As maybe a problem. And here we see methanogens here. What they actually do is they take this hydrogen gas, that buildup of the hydrogen gas, And essentially they break it down. So they're counteracting this buildup of hydrogen gas. So if we don't have a buildup of hydrogen gas, we never have that negative feedback inhibition. We can't stop the fermentation. If we don't stop the fermentation, we're continuously making more and more calories. So this is we can say our short chain fatty acids that are continuously being made as a continuous fermentation process, and that can cause us to gain weight. So I know this is a really complicated connection, so I'm just going to go over it one more time. Our body ferments food, and so does bacteria within our GI tract. When bacteria ferment the additional food, they produce hydrogen gas. The hydrogen gas builds up, and it builds up to a certain amount that it will stop fermentation. So it's what we call a feedback loop. So feedback inhibition stops fermentation because we don't need any more fermentation. We have enough of the end product, hydrogen gas, and we've made enough calories. But in people who have their gastrointestinal tract colonized by methanginins, the methanogen itself can oxidize as hydrogen gas, so it breaks down the hydrogen gas. So we never have a buildup of it. And if we never have a buildup of hydrogen gas, we never tell the bacteria to stop fermenting. If they continue fermenting, they continue making calories for your body, and these calories can lead to weight gain. So what's really interesting about this is when you hear dieting in the media, you hear metabolism, we hear calories in, calories out. Well, it's actually a lot more complicated than calories in and calories out, as we see here. Our microbiota actually is regulating how we gain weight. So looking at what kind of microorganisms are in our body might actually change how we can handle healthy weight loss. Okay. Let's go ahead and move to the genital urinary tract here. So looking at the genital urinary tract, we might recognize some of these microorganisms like lactobacillus or maybe candida here. So candida is actually a fungal a yeast. So if people have a yeast infection ever, a yeast infection can come about by dyssymbiosis. We see that the vagina is colonized by all of these different species of microorganisms. So they're living together. But the second we take something like an antibiotic, we can kill some of the bacteria, which creates more territory for the organisms to grow. So something like candida, this yeast, can grow out of control, cause a dyssymbiosis, and cause a yeast infection, so an overgrowth of yeast. So yeast infections are actually caused by microorganisms that are natural to our body. It's part of our microbiota, but it's the balance that's really important to keep them down. And that's why it's typical following antibiotics that we see kind of an opportunist microorganism that was part of our healthy microbiota taking over and causing problems by its overgrowth. Other things we're going to see here, we see lactobacillus. We're going to talk about microorganisms that can ferment later, and we're going to see how our hygiene standards have changed from thousands of years ago to today. So where we got fermented products from, kind of what microorganisms were in those fermented products, and maybe where they came from. If you want to do a little bit of research on that, feel free to. You can look up what microorganisms ferment milk into cheese, which microorganisms are used maybe to make something like wine, something like that, if you're interested. And we'll talk a little bit more about where those microorganisms came from and how did we invent things like cheese? How did things like wine come about in the first place? What interactions had to occur in order to bring these things together? So we'll talk about that a little bit later in class. Okay, has a lot to do with hygiene too. Looking at the benefits of the microbiome, again, it's a mutualistic relationship. We're benefiting from the microbiota. We talked about training the immune system, providing vitamins, providing additional calories. We have all of these benefits, and we're providing them with a home. So a safe home, essentially they're being protected from any kind of other microorganism that would want to break them down, consume them. So protection is beneficial for them. Okay. That's enough for that one. Let's go ahead and move forward. Let's talk about introtoxins. So, looking at relationships of microorganisms within our body is very, very complicated. So, while we have mutualistic relationships with them, we can also have some negative side effects as well. So, introtoxins, some of our gram-negative pathogens, can produce toxins that can cause diarrhea. So, it would definitely be a negative factor. So, the double side of our microbiome. Diarrhea can be very dangerous because it can dehydrate you to the point of death, essentially. So enterotoxins are pretty serious to look into. Other enterotoxins, though, on the other hand, can be beneficial. So some enterotoxins can alter metabolic pathways that change how cells divide. When we look at cell division, it's really important to understand cell division for understanding cancer, because cancer is hyperproliferation of cells. So, if we can increase anti-proliferation pathways, we're going to prevent cancers. So, our microbiome can also alter maybe our rate of cancer or susceptibility of cancer. And this can change throughout our lifetime if our microbiota can change. Okay, talking about cancers, we can talk about colon cancers. Colon cancers are the second leading cause of cancer deaths related in the US. Personally, my work in biotechnology, I worked on colorectal cancer. So this hits home to me covering this topic, and I wanted to bring in a little bit of my biotech research for this one. I've worked at two biotech companies in the past. I've worked at Introgen and Roche. When I worked at Introgen, it was just in these startup phases, and I was able to run specific experiments where we were looking at biomarkers for cancer. You may recognize some of these here, like KRAS, NRAS, there's EGFR, BRAF, if you've ever heard of these before. There's specific biomarkers or specific genes that are mutated that are found to be associated with cancer. So in the assays that I worked on developing here, where we were spearheading assay development, we would identify these specific mutations through TAC-MELT assays. And finding these specific mutations in genes, whether someone has an NRAS or BRAF mutation or both, is going to change their treatment. So we would run assays, and these lines here is a PCR assay we'll talk about later, where we can see the identification of specific biomarkers once they pass a specific threshold. So we can take a human sample and we can identify which genes are mutated in order to address which kind of chemotherapy treatments would be best for them for the highest survival rates. Yeah, so it was colorectal cancer. Okay, so looking at our microbiome, again, we have opportunistic pathogens that can be problematic. And this is mostly problematic in immunocompromised hosts. Our immune system can keep our microbiota in balance. If our immune system fails to do that, this is when opportunistic pathogens can begin an infection. Okay. Looking at dyssymbiosis, again, this is altering the balance between our microbiota. So just like we said, something like antibiotics can actually cause dyssymbiosis. So it changes the balance of our microbiota. We can take probiotics to help with any kind of dyssymbiosis here. So probiotics can help restore a natural, healthy microbiota. So mixing up all my slides here. A lot of times after antibiotic treatment, we can have GI issues like diarrhea or vomiting, and that can be associated with dyssymbiosis. So we've killed a good percent of our own natural microbiota along with the bad microbiota. So we take probiotics to restore the natural, healthy microbiota that we had before. Okay. Moving forward, we want to look at how our microbiota can influence our host itself, or actually our microbiome, we can talk about in this case. Looking at the microorganisms that we have living within us, they can actually alter our gene expression. So we said they can change our hormones, they can train our immune system. Well, they can alter gene expression, our own gene expression, so they can communicate with our body. So the depth of interaction between our body and microorganisms is really unknown, and it's a new field. And we're going to talk more about the gut-brain axis in just a bit too. Okay. So let's look at the blood-brain barrier. When we look at spinal fluid with the gut-brain barrier, or sorry, the blood-brain barrier, we know that these kind of fluids in our body should essentially be sterile. There shouldn't be microorganisms within our blood. There shouldn't be microorganisms within our spinal fluid. And specifically, we don't want microorganisms going up to our brain. And this is where the blood-brain barrier is. So, with the blood-brain barrier, it's a barrier preventing microorganisms from moving into the vascular system that feeds the brain blood. So here we see these tight junctions, and these tight junctions, as seen here, even closer up, are so tight that even microbes cannot fit between them. So this prevents microorganisms again from going to the brain and causing disease. So we have diseases like meningitis. It can be viral, fungal, bacterial, which is when we do get microorganisms within the spinal fluid itself and around the brain. Okay. Let's talk about the placenta also. So, something unique about the placenta is that it's going to help separate out the blood of the mother from the blood of the child. So, we have the fetal blood circulation and maternal circulation that's separated. If we look at this here, we see these capillaries where there's diffusion. So, we have the maternal circulation blood here and fetal circulation here. So, they're not actually connected. And so, the blood of the mother is not going directly into the blood of the child as we see. So there's capillary diffusion. So things that the mother has in her blood can diffuse into the child, but it's not directly her blood going into the child itself. So this helps protect the child as well. Okay, looking at our microbiome, we can look at our gut-brain axis. So something really interesting, if we look at the colonization of microorganisms within our gut, which is one of the most diverse microbiomes, we see that the microorganisms can actually communicate essentially with our brain through the vagus nerve or through these spinal nerves and communicate with us. So we see that we can have influences on our stress, anxiety, and mood behavior from our microbiome. So not only can our microbiome, again, help with our immunity, not only can it change our weight gain or weight loss, it can also affect our mood, our behavior. So that's pretty interesting to know. The more and more we're learning about our microbiota and the microorganisms that we live with that outnumber us, we're learning that they shape a good part of our life, a good part of our health, whether it's mental health or physical health, as we see here. Okay. So the last thing I want to cover is a FMT, a fecal microbiota transplant. So when people have severe gastrointestinal disorders or gastrointestinal distress, where it is compromising their livelihood, maybe they have continuous diarrhea, these kind of things. So severe cases of gastrointestinal disease. One option that's considered is a fecal microbiota transplant, an FMT, which is actually the fecal matter from a healthy individual. The healthy individual's fecal matter contains their healthy microbiota. This is put into pills or it's put through an oral tube down to the stomach, and it's transferred from one human to the next. And so the patient is the one with the gastrointestinal disease, gastrointestinal distress. It receives this fecal microbiota transplant, and hopefully that bacteria colonizes their gastrointestinal tract and gives them a healthy organization of microbes. So previous to that, they were probably suffering from dyssymbiosis. They had microbes that were not beneficial to them, causing diarrhea. With the new transplant, essentially, of the microbiota, they're able to gain a healthy microbiota within their gut. So just like an organ transplant, if we had a kidney that was non-functional, we're essentially considering our microbiota, our gut microbiota, like a non-functional microbiota that's unhealthy. So that's where we're getting the transplant from. And I know that a lot of us are going to think that that's really strange, but if we were in the situation of severe gastrointestinal distress, where it's threatening your livelihood, where you couldn't leave the room, where you were continuously hospitalized, I think most of us would consider the FMT, the fecal microbiota transplant at that point.